everyone, I'm Arya. I will now introduce the MAPK pathway. MAPK stands for mitogen activated protein kinase. On the cell membrane, the ligand FGF binds to FGF receptor, a type of RTK. The RTK dimerizes and autophosphorylates. GLB2 binds to the RTK via the SH2 domain. SOS then binds to SH3 domain on the GLB2. GLB2 and SOS phosphorylates RAS GDP into RAS GTP. This active RAS activates RAF, triggering a kinase cascade. RAF phosphorylates MEK, which then phosphorylates ERK. Activated ERK now leaves the complex and moves towards the nucleus, where it activates TF. This leads to cell proliferation and cell survival. Nitisha will now cover the physiological role of the pathway.
Cell proliferation. The phosphorylated transcription factors lead to the expression of genes that promote cell cycle progression and production of cyclin and CDKs, which facilitate the cell cycle. Overall, the MAPK pathway integrates growth signals to regulate cell proliferation. The second role is cell differentiation. TF promotes the expression of genes that lead to specialized cell functions and characteristics, guiding the cell towards a differentiated state, such as muscle, nerve, or other types of specialized cell. The third role is survival and apoptosis. For cell survival, the ERK pathway is initiated by growth factors, promoting survival by activating anti-apoptotic genes and inhibiting pro-apoptotic proteins. This helps cells survive stress and promotes cell cycle progression. And for apoptosis, the GNK and P38 pathways are triggered by stress signals like UV damage and activate pro-apoptotic genes, leading to programmed cell death. The last one is inflammation. Inflammatory cytokines like TNF-alpha and IL-1 activate receptors that initiate the MAPK pathway, particularly P38 MAPK, and amplify the inflammatory response to various stimuli. Next, Mika will be discussing about the possible malfunction of the pathway.
The most common malfunction in the FGF-ERK pathway is the mutation of the RAS protein oncogene. Typically, RAS activity is controlled through dephosphorylation by the GAP protein. This mutation prevents the GAP protein from dephosphorylizing RAS, keeping the RAS active. Due to the downward cascade of this process, the active RAS will continually stimulate phosphorylation of all subsequent factors, eventually leading to uncontrolled cell proliferation and eventually cancer. These cancers include, but are not limited to, liver, breast, pancreatic, colorectal, and ovarian cancer. And now Jeremy will discuss some lifestyle changes to reduce the contraction of these cancers.
Increasing intake of unprocessed plant foods and cutting down on high-calorie foods, as well as increasing physical exercise, reduces the risk of obesity, which causes a rise in insulin and IGF levels. IGF encourages inflammation, blood vessel formation around a tumor, and discourages cell death. Insulin also activates the RAS MAPK pathway, encouraging cell proliferation and protein synthesis. Alcohol is also known to trigger the RAS MAPK pathway, so cutting down on alcohol would help as well. Now, Kumara will talk about one drug therapy against RAS-related cancers.
A recent drug, sotorasib, relies on the cysteine to bind at the 12th position. This binding is selective and irreversible, inhibiting the allosteric site. Hence now, the particle prefers GDP more instead of GTP, making it inactive and unable to activate RAF.
Thank you guys for watching.
If you were enlightened by this video, please leave a like.